Okay, hi class. We're moving on to chapter 14 now, and this is biotechnology and industry. And these are some real practical ways in which the microbes can work for us. So after reading this chapter, you should be able to distinguish between primary metabolites and secondary metabolites, discuss the industrial role of microbes in the production of antibiotics, vitamins, yeast, and biofuels. Describe how bacterial cells are genetically engineered to produce a specific product. Compare and contrast traditional vaccines and differentiating them from genetically engineered vaccines. Describe the use of biotechnology in the diagnostic lab. Discuss the use of metagenomics. Okay, so why in medical circumstances we need to be able to sequence a genome. So a genome means the total DNA of the microbe, um, and of course we're then by finding the sequence of all the DNA, we're finding all the sequence of all the genes that are in that microbe. Okay. And one of the things that a microbe can, or the genome can tell us, is um, what genes are possibly there. Okay, and so in this example of the Streptomyces collicolor, um, we're looking for a gene that's going to be able to produce some stains, um, and uh, usually something like a pigment is not just a simple one gene, one product. You're going to have a whole metabolic pathway that you have to sort of dissect from the DNA sequences. Okay. Um, Something that's very true, and especially with streptomyces, um, things like antibiotics. Okay. Now the antibiotics are a byproduct of the bacteria that's producing them. So not like the bacteria were evolved to make the antibiotic, but here we can have this pathway, and we can try to find new antibiotics. So hopefully we can cure human diseases with them. And another thing that's really important here is we can see the relationships between different microbes. So out in nature, different microbes are going to be living together, uh, and by doing a metagenomic approach, we're going to be able to see which bacteria are there, and then we can kind of figure out how they all work together. And I'll just kind of tell you that one of the places where we don't understand enough, we just need to do more of this sequencing, um, is in the mouth. And so there are lots of bacteria in your mouth, and we can probably treat a lot of disease of the mouth um, by being able to dissect out how the different microbes are relating to each other. Okay. So in industrial microbiology, we use microbes uh, that naturally produce desired products. Um, so that could be an antibiotic, it could be a starch. It doesn't matter what it is. The industrial side of it is that we are using the microbes to produce a desired end product. Okay. Now in biotechnology, now we're going to use some genetic engineering to alter the microbial genome so that it can produce what you want it to do. Um, Since E. coli is sort of a lab standard and almost anyone can grow E. coli, one of the things that we sort of often do is we might take a metabolic pathway from some other bacteria that might be really difficult to grow and then put the genes into the E. coli and then be able to use the E. coli uh, to make that product. Okay, and of course, then we can grow the E. coli quite easily, and hopefully then switch it into a sort of industrial microbiology setting where we can make as much of that product as we want. Okay. One of the big pluses being done right now is human growth hormone. Um, and human growth hormone is a protein, and proteins are hard to make just in the laboratory, just from scratch, making them in the laboratory. Um, so if we can take the genes from a human and put it into E. coli, then we can grow up the E. coli and make as much of the human growth hormone as we want. And then people who have hormone deficiencies, we can give it to them so that they can grow properly. Microbes in industry. Uh, Prokaryotes and fungal microbes are uh, metabolic specialists, and in a way that makes sense. They are things that can grow independently. They don't need uh, some other cell type to be able to grow. Um, so all these microbes are going to have their uh, all the specialties they need to have all the metabolic reactions that they need. Okay. Just in contrast to that, if we were talking about using human cells, we would need to be able to support the human cells in other ways so that we could get the human cells to be able to make whatever we want them to make. But we don't need to do that with a microbe. Okay. Um, so we're going to make end products called metabolites, and we're going to be able to produce high quality of the metabolites in a short period of time. And that's why it's an advantage to use a cell maybe like an E. coli, something that has a relatively short half-life. That is, it doubles every 20 minutes. And so we can produce a lot of those kinds of cells, and then those cells can all make their secondary metabolites, and we can then harvest the metabolites. Okay, like I said, they grow rapidly, um, and we can some depending on the bacteria you use, you can get spore production. Something like Bacillus subtilis will definitely produce spores and is relatively easy to grow in the lab. Something like E. coli doesn't make spores, but still very easy to grow in the lab. Um, a nice thing about using industrial microbes is that these you can engineer it so that these are not animal or human pathogens. Um, we wouldn't want to have to uh, make a whole big vat of human pathogen because the workers who are working in that factory would be at risk of getting the pathogen. Okay. Um, Primary and secondary metabolites. So, a primary metabolite is something that is normal to the growth of the microbe. Okay, and so it's just used in sort of a normal growth mode. And the um, primary metabolites are generally produced in log phase. Okay, so if you remember back to the growth lecture, the log phase is when the cells are increasing logarithmically. Okay, so it's kind of a constant um, growth. The cells are dividing pretty reproducibly, and that's where you get all your primary metabolites. Now, your secondary metabolites are not essential for growth, and these sometimes tend to be a little bit stranger things. Um, and they are only made from the very end of log phase into stationary phase. So while the cells aren't dividing, these cells are really good at producing these secondary metabolites. Uh, we know in the case of most of the yeasts that the secondary metabolites are almost always produced during that non-growing stationary phase. So industrial fermentation. So any procedure with or without oxygen used to grow a large number of microbes, um, and you're using specialized fermentation tanks. Um, Industrial microbiology, the products. So one of the major things that we're going to be making is antibiotics, and penicillin is one of those antibiotics. Um, and these would be secondary metabolites. Okay, so uh, the molds that are making these don't need the antibiotics to survive, but they're going to be able to kill pathogens, and that's again a secondary metabolite that just happens to be being made by that mold. Okay. Um, these are natural products, and we can make a number of synthetic derivatives. And as we talked about in our discussion of penicillin earlier in the semester, um, we can have a number of derivatives that just substitute something out that beta-lactam ring. Um, I won't go into Fleming's discovery of antibiotics. Okay. And there are several natural forms, but the synthetic forms are generally what are being used today. We can use uh, the industrial microbiology process to make a number of vitamins. And things like B2 and B12 are definitely things that are added to food products all the time. And you want those things in your food products. Okay. Uh, and these are these vitamins are second only to antibiotics in terms of sales. Um, and there are so many things that are using vitamins, um, especially as food additives, um, that these are big producers. Um, and these are generally vitamins that the human body can't make. Uh, B2, B12 are two examples of those where a human just can't make them. Um, so the riboflavin B2 is a product of Ashbia gossypii, and it's again in many foods as a dietary supplement. Cobaltamine, made by Propionibacter, Pseudomonas and Streptomyces species, and are definitely used to fortify different foods. As you look at almost any food, commercial food, you can look at the label and see that there's a large amount of B12 added. Um, Production of amino acids. Uh, Glutamic acid can be produced by Corynebacteria species, and these uh, or this can be used in the production of monosodium glutamate, which is a flavor enhancer. And unfortunately, there are some people who are allergic to MSG. Okay. Lysine produced by another or the same species of Corynebacterium, uh, and the lysine can again be added as a dietary supplement. The last two uh, amino acids, aspartic acid, and phenylalanine, produced by Propionibacterium, Pseudomonas, and Streptomyces species. Okay. Um, the aspartic acid and phenylalanine are used in the beverage industry. Um, Usually we think of those in terms of artificial sweetener drinks where the aspartic acid and the phenylalanine are combined together to make some very sweetening, great sweetening product. There are a number of enzymes that are made by industrial microbes. Um, and just as a refresher, remember an enzyme is anything that's going to catalyze a chemical reaction. Uh, in microbes, we can have things called exoenzymes, which means they're enzymes that are exported from the cell. Um, and the primary one um, among those is amylase. So amylase is produced by Bacillus subtilis and Aspergillus niger, and uh, what amylase does is break down starch into simple sugars, generally right to glucose. Okay. And these can be used in beer making, dough production, uh, removing spots from clothing. Um, and you can also use this to make high fructose corn syrup, um, and that's a very productive because so many beverages, especially, have lots of high fructose corn syrup. Okay, so protease is something that breaks down proteins into peptides, and again, things like in bread making, laundry products, meat tenderizer. The meat tenderizer is one of the very prime uses, um, and you can you can buy it commercially in a grocery store. Any kind of uh, shake on um, meat tenderizer is probably going to have some proteases in it. Okay, so renin, that's the enzyme that's going to curdle the milk for making uh, cheeses. organic acids. So citric acid is an acid that we use a lot of, and that is produced by the mold Aspergillus niger, and it can be used as a preservative or as a souring agent in soft drinks, candies, or wines. Um, and again, sort of a more natural way of producing a sour flavor um, by the use of citric acid. Lactobacillus are going to produce lactic acid, and we can use those as either a flavoring or a preservative. Now, biofuels is sort of an up and coming use of industrial microbes. Um, so we can make fuels out of some renewable biological material. And one of the things that's very often used, something uh, like leaves, plant leaves, or in an extreme case, you might go to a sugar cane. As long as you can grow a lot of that material, and that material doesn't have some other economic benefits, that's where you run into the problem with this sugar cane. The sugar cane, the sugar part is also quite valuable. Yeah. Um, so in this country right now, a lot of vegetable oil is being used uh, to turn into biofuels, and one of the ways that this is done is vegetable oil used from uh, something like a McDonald's. Once they have to drain the French fries, the oil from where they're making the French fries, we can use that. And once it's been used to cook the food, then we can turn that into a fuel. Um, the sort of easiest way and most productive thing to do is to just produce ethyl alcohol or ethanol. Um, and again, in the previous lectures, we've talked about uh, how to produce alcohol from sugars. Okay. Um, when we're combining uh, naturally um, produced um, oil with some of the alcohol, then we're getting gasohol. Okay. Um, we can also use a lot of algae using algae cultures. Um, so the algae is the source of the oil, and the algae are actually producing little droplets of oils, um, and we can just convert those oil droplets that the algae are producing into a oil that we can use for driving a car. The industrial industry is having some problems with scaling up this process because right now we can make small amounts of the biofuel, but where we start getting into real problems is when there's just not quite enough uh, of the fuel being produced by the algae. Microbes and gene technology. seen the promise. Okay, so genetically engineered bacteria means that we've taken some of the DNA from another cell type and put it into the microbe that you're looking at. Um, and this can be done several ways, specifically to make human proteins. It's a very good method. Um, and Human growth hormone, HGH, is one example of that. Since human growth hormone is a peptide, uh, having a bacteria produce it is among one of the best ways to do it. Um, so to do that, you can take the gene sequence out of a human, and make the very small piece of DNA, and then clone that into a plasmid vector, which is a small circle of DNA that a plasmid usually carries, and then you can put that gene in there, making the hybrid, and then introduce that plasmid back into a bacteria, and then that bacteria will make the human growth hormone. Um, on a very practical note, I myself actually use insulin to make human insulin, and that is made genetically in bacteria. So hopefully, um, it's a good treatment for diabetes, and is really effective because it is real human hormone. So we can also make proteins that we can be using for vaccines, or actually make synthetic vaccines. So in sort of the old technology, the way you make a vaccine is to give some preparation um, of uh, antigen or part of the virus that won't uh, will only make the immune system uh, hype up, but won't actually make you sick. Um, so one of the ways of doing that is to take the virus and kill it with something like formaldehyde or glutaraldehyde, and make it so that it can't replicate, but yet it still has the same shape, and so the immune system can recognize that shape. And in that way, you're going to be able to generate immunity. So, and you have to have booster shots, but you take a little bit of it, and that's going to prime the immune system to know to look for that if it is ever exposed to it. Now, one way or another way that you can make these is to use attenuated viruses. Um, and in attenuated vaccines, you have a weakened pathogen, and they just don't go on to cause disease. Um, these are generally mostly pluses, but there could be some minuses. Um, they may not work quite as effectively, or they might not have been fully weakened, and so maybe they'll cause disease in the human. Um, now, one of the best things that's going to happen with a attenuated vaccine is that you're going to generate a very large response. And one of the problems with that is you need to refrigerate that vaccine, and so not really a problem in this country, but in many parts of the world where it might be days or you're near a refrigerator, you really can't use that vaccine there. Inactivated vaccines um, contain the pathogen but can't reproduce, and you can't get the disease from that vaccine, uh, and can generally be stored at room temperature, which again is a huge advantage if you're going to a part of the world that has no refrigeration. Now, another minus is that this is often going to generate a weakened immune response. Uh, these inactivated vaccines are just not quite as effective at making good vaccines. So in toxoid vaccines, now you're trying to get a vaccine um, just to the toxin that the bacteria is going to generate. And you can't get the disease because you don't have the organism at all. You only have the toxin present. Uh, this generally requires many booster shots um, because the exoenzyme is going to break down in the body, and so you need to give a lot. You need to give a lot of it over uh, a long period of time, so that you're keeping an immune response to that toxoid. Um, Now, if you use a genetically modified, okay, um, you can actually just clone one part, one subunit of the vaccine, and use that to mimic the pathogen and not even have the pathogen present. Uh, these are in general pathogen proteins, and like I said, it's impossible to get the disease from these synthetic genetically engineered vaccines because the whole organism isn't even there. Only part of the organism is there. People are trying to develop DNA vaccines. Come in.
Hello.
Hi.
Hey, Bill. I have your medicine. And it's your 10 o'clock.
Okay.
Where are you going to put that? Right here? Okay. Thank you. And I have to check your blood sugar. If you want to check it, you can. Whatever.
You can fill it all in. Okay.
Thank you. Okay. Did you want to do a shot or you want me to do it?
I can do it.
All right. I'll write it down.
331 is 8, right?
331, I think 8 or 10. Oh, I got the chart right here. 16, 8. All right, thank you, Bill. Yeah. Is this working out for you like this? Yeah, it's fine. Is it all right with you? Yeah. Okay. I'll see you at 10. All right.
Okay. The DNA vaccines are an advantage because usually isolating DNA from an organism is quite simple. Okay. And in that way, we can worry about genes that are specific to the pathogen. And then when they're injected, the body produces proteins uh, to which the immune system are going to be able to respond. Um, Again, in just the case of DNA, you can't get the disease. So in this summary, you can take uh, one of these plasmids, one of these circles of DNA, and use that as a vector, and then take the gene that you're interested in and clone those together, making a recombinant molecule. And then you can put that recombinant molecule into a second bacteria, and then the second bacteria will make many copies, and we can make lots and lots of copies of that so we can produce that protein. Um, and here's one of the examples I was talking about. Um, this second bacteria could easily be in E. coli, which again we know quite a bit about how to grow E. coli, and we know how to optimize E. coli so that we can get a lot of protein production. So how do we make probes to be able to detect different microbial uh, things? In the polymerase chain reaction, we take a single piece of DNA and then separate the two strands with heat, then add little pieces of DNA called primers, and then the enzyme Taq polymerase, and that Taq polymerase is going to make new strands. And so you can see we've now made two new strands here. Then you denature those again with the heat, and again add the Taq polymerase and the small primers, and we just keep repeating this. Generally, repeat this about 30 times, and so you can start with just a single copy of DNA and get over 1 million copies of the DNA quite easily. Um, Now, we can use that to be able to um, we could clone that PCR product, um, or we could develop some sort of array system where we have single-stranded DNA on a chip, and then we can take copies of that DNA, label them, and then allow them to float over the chip, and then they're going to bind specifically to part of the DNA. Um, Gene technology, the antimicrobial world, and altering life. So one of the things we can do is a metagenomic approach where we're trying to analyze the communities that are present. Okay, um, and one of the things that we've seen a lot of times, especially in water microbiology, is that we have these viable but non-culturable bacteria. Okay, which means that they do produce, they're metabolically active, but they're not able to grow in the laboratory. Okay, and so we can just detect their DNA, and in that way we can see who's there, even though we don't have to grow them. Because traditionally, you just grow a culture, and you just have millions of copies of that uh, bacteria, generally overnight. But here we don't even have to do that. We can just take the DNA from these cells and know that something is there, uh, and assume that it is metabolically active. Okay. And like I said, the word for that is metagenomics. Okay. And so in that way, we're attempting to see all the viable but non-culturables through sequencing their genomes. Uh, so what has the metagenomics shown? Um, and this is definitely useful in medicine, um, And one of the places that I think is really important that we haven't done yet is in oral health. Um, Also in ecology, and just trying to find out who's out there in soil. Um, the vast majority of microbes that are living in soil don't reproduce in the laboratory. Um, Possibly to produce biofuels or produce energy in some sort of way, possibly in the production of different foods. We might be able to find some bacteria that we will help us enhance crops. Okay. Uh, it's definitely true that while officially the government says we don't use microbes in warfare, we definitely do. I mean, there's just no way that we couldn't. Um, so it would be important to know about uh, these metagenomic approaches for biodefense. So we might be able to engineer new pathways. Um, that is, can we get the microbes to make some new products that we want to make? Um, and sometimes this is pretty difficult, especially like I've seen a seminar on oil production in the microbes, and while you can do it, it takes a whole lot of steps to be able to get the microbes to actually do the engineering quite well. Uh, so in this case, they're making the indigo dye by being able to produce um, or engineer in the metabolic pathway to make the indigo dye. And here you can see just part of the pathways here that we can take um, water and tryptophan and then convert that uh, into indole and then uh, convert that again and finally end up with that indigo blue. Um, and the bacteria that are producing the tryptophan, uh, they need an extra gene to be able to turn it from the indigo into the indigo blue. Okay, so what should you take from this lesson? The uses of primary and secondary metabolites from microbes for industrial microbiology, protein production of biotechnology, the vaccines available in old technology and new biotechnology, how DNA polymerase reacts and DNA probes work, what things we have learned through metagenomics, and the usefulness of pathway engineering. Um, and so the sort of thought question there is, uh, have you had any experience with any industrial applications or biotechnology implications? I gave you the example that the insulin that I use is made with biotechnology, so you may not have done it directly, but you certainly know someone, because you know me, who has used biotechnology um, to save his life.